it would definitely weaken the alpha helix. And so it's really hard to study membrane proteins if you dissolve the membrane and expect to do some structural analysis. So studying membrane proteins has been very difficult. So if we could look down the alpha helix, okay, we could just get a bird's eye view and look straight down the middle of the alpha helix. The R groups are going to be sticking out. Did any of you in organic chemistry think about the design of peptidomimetics? Ring a bell? Okay. Our biochem seminar this past spring was on peptidomimetics, and it was the design of molecules that have the same dimensions of an alpha helix, but are not alpha helices, and they're extremely stable and look like they have good drug potential. But the R groups stick out from the alpha helix. So what kind of R groups might you find in the part of the protein that spans the membrane? Probably pretty boring hydrophobic amino acids. Okay, and what I like about the book shows you, let's see if I can find that page, on page 579, it shows you some multi-pass proteins and single-pass proteins, and you can clearly identify the region of the protein that's transmembrane because the amino acids are really strongly hydrophobic. So back in the 80s, it took about 10 years to find the cystic fibrosis gene. And once they found something that had gene-like characteristics, they sequenced it, they looked at the codon sequence and imagined what kind of protein could be made by this gene. And they noticed that there's several stretches in this protein, if this happens to be the protein made by the gene, there are several stretches that are very distinctly hydrophobic stretches. So they imagine, well, maybe it is a transmembrane protein because of these distinct long stretches of hydrophobic amino acids. And they were actually correct. It was a transmembrane protein. It's a chloride channel. But they just imagined from the gene sequence what the amino acid sequence of the protein was, because they had no idea what kind of protein was mutant in people with cystic fibrosis. They knew it was probably one because of the genetic inheritance pattern, but they didn't know, had no clue. So the sequencing of a potential protein that could be the cystic fibrosis protein revealed a multi-pass protein. Okay, so that's easy to predict then if you see an amino acid sequence. We're also going to then see that this gives that there are proteins in the associated in the cytoplasm but close to the rough endoplasmic reticulum that will also detect stretches of hydrophobic amino acids. And that helps start the insertion of a protein into the cell, into the ER membrane. Okay. So hydrophobic amino acids are a really long stretch of hydrophobic amino acids are pretty good telltale signs that this is a transmembrane protein. Oops. Okay, one other picture. The alpha helix might be if there's a transmembrane protein, maybe a multipass, and these multipass proteins align themselves kind of to create a channel. Might be a sodium channel, or a potassium channel, or a chloride channel. Okay? The reason for the multiple passes is those alpha helixes will arrange themselves into a channel structure or into a structure that can accommodate whatever is being transported. So it might be chloride or potassium, maybe glucose, although glucose does not go through a channel, but okay, so there are some of the R groups okay, that are poking into the channel, and then there are some R groups that are poking into the hydrophobic tails, fatty acid tails of the membrane. So all of these that are oriented such that they are in the hydrophobic domain of the membrane, clearly they're going to be hydrophobic amino acids, valines and isoleucines, maybe some glycines. Okay? But what if it was the middle of the channel? What if it was inside the channel? And this was a chloride channel, for example. Would you want hydrophobic amino acids in the chloride channel? Would the chloride pass through it? No, the chloride channel would not. The chloride wouldn't even be attracted to it. How about some negatively charged amino acids? No, you would not want to put some negatively charged amino acids in your channel. And it is true that the cystic fibrosis channel, which is a chloride channel, has some positively charged amino acids along the alpha helices, but the arrangement of those negatively charged amino acids, positively charged amino acids, is that they would be oriented inside the channel, not associated with the hydrophobic fatty acid tail. So the presence of a couple hydrophobic or positively charged amino acids in those hydrophobic stretches of amino acids suggested that, oh, this could be an ion channel for some negatively charged ion. Okay, so it's not true that the alpha helix that spans the membrane has to be completely hydrophobic. It might have to carry some characteristics that would attract whatever it is that it's transporting. On page 581, the book shows you that not all channels are made from alpha helical structures. Okay? There are those kinds of channels that are created by native cleavage sheets. And we're not going to really cover any of those types of proteins. I think aquaporin, which carries transport to water, would fall under that category. So we're not going to cover those. Before we get down to, I want to talk a little bit about prenylation, because some of you are in the biochem lab, and I know some of you aren't, but you can still learn about prenylation. I wanted to give you a couple other characteristics of proteins. That you would find in the number. This will be our like our insulin receptor. It could be almost any integral membrane protein where part of the protein sticks out from the cell and part of the protein is sticking into the cytoplasm. Very frequently, integral membrane proteins will have some sort of carbohydrate residue attached to it. So those hexagons are sugars. They're usually called oligosaccharides. Oligo meaning a few. So if you need a stiff definition for oligo, it's anywhere from 3 carbohydrates to 10. Okay, that's what an oligosaccharide is. They're only on non-cytoplasmic surfaces. You never find the carbohydrate attached to the protein, that at least that part that's sticking into the cytoplasm. Okay. This, by the way, becomes a glycoprotein. And almost all integral membrane proteins of the plasma membrane are glycoproteins. Okay. So if you find a glycoprotein, or if you discover that, oh, here's this protein, we isolated this protein, it has a oligosaccharide attached to it, that should give you a clue as to where would you find this protein in the cell. Okay. So that's one thing that gives you some clues about the orientation of a protein that you might have isolated. Do you recognize this protein? Or this protein complex? What is it? Doesn't that look surprisingly a lot like an antibody? Yeah, so this is a typical, now see, but it's an antibody structure. This happens to consists of 4 different proteins, and how are they attached to one another? How are they anchored? By disulfide bonds. And where are the disulfide bonds? They're on the non-cytoplasmic side of the cell. Okay? So here's another thing that you would only find on the side of the membrane, that's the non-cytoplasmic side of the membrane. You might find disulfide bonds. Never disulfide bonds in the cytoplasm. This happens to be a reducing environment. Okay, so it favors cysteine residues that have the sulfhydryl groups, that the hydrogen remains attached to the sulfur groups. That means that no disulfide bonds form. This is an oxidizing environment. Which means that it favors the loss of the oxidation of the sulfur, and that creates the disulfide bond. The disulfide bonds are going to form in the endoplasmic reticulum. That's the job of the endoplasmic reticulum, to create the correct combination of disulfide bonds. Okay, so that's something else. Okay, have you ever taken omeprazole, or what is it, Prilosec? I think they're the same thing. Okay, the drug omeprazole forms disulfide bonds with the proton pump, and that inhibits the function of the proton pump. Okay, so where do you think you're going to find, did that give you a clue as to where you might find the omeprazole bound to it? Is the omeprazole bound to the hydrogen pump on the inside of the cell or the outside of the cell? The outside of the cell, yeah. And I think the omeprazole becomes disulfide bonded in the ER, in the endoplasmic reticulum, because the drug has to go into circulation and into cells. So it can't just go into your stomach and disulfide bond to the hydrogen pumps in your stomach, which I used to think. But apparently it needs to be absorbed. So my hunch is the inactivation of the proton pump happens in the ER as the proton pump is making its way to the plasma membrane. Oh, I only get about 5 minutes.
Why is that receptor inside the membrane? The insulin receptor, why is it inside?
Why is it?
The insulin receptor.
Oh, it has a cytoplasmic domain that happens to be an enzyme. Okay, tyrosine kinase. You guys heard of tyrosine kinases? Yeah. And that's how it does its effect. So insulin binds to the insulin receptor, okay, to activate the tyrosine kinase activity, and it's this tyrosine kinase activity that then does all of the effects that insulin would normally do inside the cell. So, like all water-soluble hormones, the binding of the hormone to the receptor activates some domain down here on the cytoplasmic surface that then initiates the changes in cell physiology.
So insulin is, the receptor is actually outside, not inside.
It's both. Okay, the receptor binding site is outside the cell. The receptor binding site is outside the cell. And almost always, insulin will not get into the cell. Okay, experimentally, you can force insulin into the cell, but the insulin that when it's brought into the cell will be degraded. So when I first started teaching cell biology, we thought that insulin had to get into the cell in order to change the physiology of the cell. And what a silly concept, you know. Now that we know how insulin works, like who thought that? So, okay, so I think what I want to talk about then, not today, but we'll do it tomorrow, is I want to look at prenylation of key proteins, and I want to look at the mobility of proteins in the membrane, because we know that lipids are fairly freely mobile, at least in the apical side of the cells. The lipids can go practically anywhere. On the basolateral side of the cell, the lipids can go anywhere. But what I want to look at is the mobility of proteins in the lipid bilayer. And there's a classic experiment that was done on page 589. You'll have to read the caption. The experiment was done in 1970. It's a classic experiment. And we'll talk about why that experiment worked for that kind of cell and why it doesn't work for all cells. Okay. Then maybe we can get out early to transport. I think